Biotechnology Principles and Processes. The second lecture in this series, we will talk about the first two steps of the process of biotechnology. In the first lecture, we have already discussed about the principles and the basic principles involved, as also the processes in brief. In brief, we have talked about. Now we will move into the details of each of the steps. In the second lecture, we will talk about the first two steps mainly. The processes of recombinant DNA technology, just a recap of what we had already talked about previously, so that it becomes easier for you all to follow. So in the processes, it involves, first is to isolate the DNA and plasmid from the cell. From the cells or the organisms, the DNA, whichever DNA we want to work with, the gene that we want to extract, as also the plasmid that will be used as the vector, we have to isolate that. That means we have to take it out from the cell by using some digestive enzymes. The second step, which is after we take out the DNA, it has to be cut out. It has to be cut into small fragments using some enzymes, that is restriction enzymes. As also, we will apply gel electrophoresis to separate out the fragments, and followed by polymerase chain reaction. Then next is ligation. That means we will join the isolated DNA with the vector, followed by transferring of the recombinant DNA into the host cell, and finally obtaining the product with the help of bioreactors. So the steps which involves that is extraction of the plasmid, that is the DNA, as also the DNA that has a gene of interest. We will isolate it out using certain digestive enzymes. The first step. The second step will be the next step, which involves cutting of the plasmid, as well as so that to create a space, as also to cut out our gene of interest, the gene that we want to be transferred. So the second step involves that, and followed by ligation and then transferring. So in the present lecture, we will talk about isolation of DNA and the plasmid, how it is done with the help of digestive enzymes. We will learn that in details, as also we will learn the cutting or fragmentation of DNA at specific location using the restriction enzymes. The how the restriction enzyme actually works, that will be talked about in details. Now, isolation of genetic material. Now, how is the genetic material isolated? Of course, prior to isolation of the genetic material, we have to identify the gene. That means we have to recognize the gene, the DNA, which contains which is capable of producing our desirable traits or characteristics. We want an antibiotic now, or an antibiotic resistant we want. So what we will do first, we will have to track out that which is the specific gene creating that particular, giving us that particular characteristic. So that is identification. After identifying, what we'll do, we will isolate that particular genetic material. How? From the cell. The cell, it might belong to the DNA. It is to be isolated in pure form, either from a microorganism, or from a plant cell, or from an animal cell. So from any of these organisms, the DNA has to be isolated in pure form. So for that, what shall we do? We will select out that particular cell, be it from a plant, be it from an animal, or a microorganism. So whatever it belongs to, we will take out that cell first, or we will take that tissue from that organism, and then we will carry out lysis. Lysis means breaking down. We will break down the cell. So what will be done? The identified cells will be treated with either lysozyme. Lysozyme is an enzyme that is capable of breaking down the cell wall of bacteria. So if our identified cell that means our gene of interest, desirable gene, is present inside a bacteria, then what we will do, we will apply lysozyme to the cell. Thereby what will happen? Consequently, the cell wall of the bacteria will simply dissolve. If suppose our desired gene is present inside a plant, then we will use cellulase. Cellulase will dissolve the cell wall of the cellulosic cell wall of the plant cells. If suppose our identified cell is inside a or it belongs to fungus, then we will use chitinase, because chitin makes up the cell wall of fungi. And if it is an animal cell, not required, because they do not have any cell wall. So cell wall is not present in animal cells, so this particular step will be skipped if it is an animal cell. But in case of bacterial cell, we will use lysozyme. Plant cell, we will use cellulase and then chitinase, followed by applying protease and ribonucleases. First, after the cell wall has been dissolved, what shall we add now? Next, the enzyme protease will be added. Thereby, what will happen? All the protein component present inside the cell organelles or anything which is made up of protein inside the cell will be dissolved, including the plasma membrane. So that will be dissolved. Then adding ribonucleases will dissolve the ribonucleic acid, that is the RNA will be dissolved within the cell. So what do we get now? We have got that almost everything has been dissolved in the cell, leaving behind the DNA, the purified DNA. Only the DNA will be left behind now. Now this DNA, what will it be done? It will be precipitated with chilled ethanol. Now that particular DNA, that means whatever extract is left behind, of course it will not be visible to us as DNA. It will remain as a solution. So what will we do? It will be added to chilled ethanol. Chilled ethanol will precipitate it. The DNA will get precipitated. It will form like a froth. It will form like a froth over that solution. Now here the steps are shown in the form of a figure, which will make it easier for us to understand. So what will be done? First is sample preparation. Sample preparation means the particular tissue is extracted out. Suppose you want to extract the DNA from onion plant cell. Okay, so what will be done? We will first prepare the sample. We will crush the sample. Okay, we will make a slurry of the sample. We'll make a slurry of the sample, that is lysis, after which we will add the enzymes, components containing the enzymes cellulase, because what did I give the example? Onion, which is a after adding cellulase, what will happen? The cell wall will be dissolved. Expected, the cell wall has dissolved within this tube. Then after which protease will be added, that will remove the protein, followed by ribonucleases, which will dissolve the ribonucleic acid. Then followed by what will we add? We will add chilled ethanol. That will lead to DNA binding. Okay, then washing, and finally elution. Elution where stains are added so that it becomes visible to us. Now in the final form, finally what will be done? We will spooling. This step is known as spooling, whereby we will insert a rod-like structures here as shown here into the tube, and then it is shrilled. In a shrilling motion, what will happen? The DNA will be extracted out, because it will be floating. It will form a precipitate, as told already. In this is chilled ethanol, so it will form a precipitate and that will be extracted out. The DNA will be extracted out. So here we have got the extracted DNA. The genetic material has been isolated out from the solution. So this is the first step of recombinant DNA technology. Now let us move to the second step. After this, what? After this is the complete DNA of the cell. But we don't want the complete DNA. We want only a small part of the DNA containing our desirable gene. That part which can carry or which will give us our desired characteristic. So what shall we have to do? We will have to cut the DNA at specific location. Specific location here means the specific gene that we want. What will be used for it? To cut out at specific location will be used the enzyme restriction enzyme. They're also nicknamed as molecular scissors. Now these restriction enzymes, they are actually present naturally in some bacteria. Naturally, they are present in bacteria as a defensive mechanism. Defensive mechanism, that is, some bacteria, what do? They produce these enzymes, which we call as restriction enzyme. That enzyme can cut DNA. So, if another organism is attacking a bacteria naturally, I'm talking about how it happens naturally. So, another bacteria, second organism is attacking the bacteria. Bacteria will release the enzyme, and that enzyme will cut the DNA of the second organism, of the parasite or the organism that is trying to attack it. So thereby, what is the bacteria doing? It is defending itself using the enzyme. So that very principle has been exploited. We have used that in cutting DNA. Okay, we have used that in cutting DNA, but naturally bacteria use it as a defensive mechanism. Here, of course, we are not making the bacteria use it as a defensive mechanism, but we are applying it to cut DNA at particular locations. Now, the restriction enzyme can be of two types: endonucleases and exonucleases. Now, what happens? Endonucleases is capable of cutting DNA within the fragment. Within the long DNA fragment, it is capable of cutting it in between. Exonucleases from any end, open end, it is capable of cutting. So here you can see activity of the nucleases. Okay, this is the total DNA that is shown here. If it is an endonuclease, it will cut DNA in between. If it is an exonuclease, it will cut at one end. See, here it has cut it, so you get this separated out. Here it has cut it, so you get this separated out. So from ends, okay, nucleotide is removed from the ends and it is internal cut in case of endonucleases. Then where will the restriction enzyme walk? It is not that it will cut at any location. It's not that. The restriction enzyme can walk only at particular points, and those particular points are referred to as restriction sequence. That means you have the DNA, you add the enzyme will look for some particular point where it can bind. That enzyme substrate complex has to be formed. So at particular points only it can form that complex. That particular point is referred to as restriction sequence. That is referred to as restriction sequence. What characterizes this restriction sequence now? This restriction sequence is now characterized by palindromic site. Now, what is this palindrome? Now, palindrome is a sequence. In English language even we get it. Palindromes are some sequences, some words which reads the same in both the directions. Like say for example the word madam, M-A-D-A-M. So if you read this word from backwards, M-A-D-A-M, it reads the same. So such words are called as palindromes in English. So similarly in DNA sequences also some palindromic sites are present. Now but what it is? Okay, in DNA sequences, if the complementary bases reads the same, like say here for example GAATTC is the sequence in one of the strands, okay, the strand that is 5 to 3 direction, and in the complementary strand, okay, which is 3 to 5, see here GAATTC. So it reads the same, it's forward and backward, it reads the same. Both these two both the sequences. Only at some specific locations within the DNA it can be found. Such sequences, when it reads the same, then it's called as palindromic sequence. So here you can see genetic palindromes are similar to verbal. A palindromic sequence in DNA is one in which the 5 to 3 base pair sequence is identical on both the strands. So 5 to 3 direction is the same, GAATTC. In the other direction 5 to 3 GAATTC. So it reads the same. So those are called as palindromic sequences, and such sequences are recognized by restriction enzymes. They will recognize and then show their action at these locations. So let us see furthermore. Now, example of restriction enzyme, HIND-2 and EcoR1. These are examples of restriction enzyme, names of restriction enzyme. Now, how palindromic sequences and the restriction enzyme gets attached, we will see that. Okay, but before that, we have to learn a little more on restriction enzymes. So let us see that. These two are examples. Now, how are they named? What is the principle behind their naming? Let us see. So here, nomenclature of restriction enzyme. How are they named? After the bacteria which produces them. Because restriction enzymes, as I already mentioned, they're produced by bacteria. So which bacteria is producing, depending upon it, it will be named. So here, genus Escherichia. That means Escherichia coli is producing particular type of restriction enzyme that we have extracted out. So from the genus, E has been taken. You can see here the name of the enzyme is EcoR1. So E has come from the genus name, the first letter of the generic name. Then the second two, CO, ECO, CO has come from the first two letters of the species. Then R, the strain of the bacteria. Then 1, the order of isolation. That means this was from Escherichia coli, this was the first restriction enzyme to be isolated out, to be studied. So accordingly the name came EcoR1. Another type of enzyme, another type of restriction enzyme, HIND-3. HIND-3, the name comes from the enzyme is extracted from the bacteria Haemophilus influenza. Now Haemophilus influenza, so H comes from Haemophilus, IN comes from influenza, the species, first letter, D from the type of strain, and 3, that means it is the third enzyme to be isolated from Haemophilus. So HIND-3. Similarly, BAM-H1. BAM-H1 extracted from Bacillus amylo, so B from Bacillus, AM from amylo, H the strain, and 1 the order of isolation. So accordingly, the naming of restriction enzyme is done. I hope this is clear for you. And these are given the restriction sites. Restriction site means, as I was telling here, the palindromic site. Okay, these are the restriction sequence. That is, all restriction enzymes are specific. EcoR1 will recognize only this type of restriction sequence, palindromic sequence. And wherever, in whatever DNA it gets this sequence, EcoR1 can cut it. HIND-3 can recognize this type of sequence AAGCTT, and BAM-H1 can recognize GGATG. So they are specific, very, very specific. What type of DNA, from where it has been extracted, irrespective of that, if the sequence is present, then EcoR1 will act. If the second sequence is present, HIND-3, this one, the BAM-H1. Accordingly, we have many types of restriction enzyme, which has been already isolated now, since the time it has started. Then here, the small arrow that is given here, this is the point where it actually acts. Okay, you should not get confused here. This is nothing but this is the exact point where the enzyme will work. Now let us see how the enzyme can cut the DNA. Action of restriction enzyme. Now we have a DNA. Okay, here the two DNAs separately we have shown, because already it was told to you previously. I have already told that we have to use the same restriction enzyme to cut the DNA in the plasmid as well as in the foreign DNA. Foreign DNA means the DNA from where we are going to extract out our gene of interest. So here this is the foreign DNA, black represents the foreign DNA and the red one represents the plasmid DNA, the vector DNA. We have to use the one very important characteristic that should be looked for while identifying is that both vector DNA that we are selecting and the foreign gene that we are selecting should be having similar types of restriction sites or recognition sequences. Only then we can use the same enzyme, and it's very, very important that they should be the same restriction enzyme should cut both of them on the similar types of same type of restriction sequence. I hope you have followed this. These two are separate cases, okay? These two are separate. This is another DNA, this is another DNA. For the red one we will use for a ligation, and a foreign DNA, that means we will use that desirable gene, that is it contains our desirable gene. So now both of them, they have the sequence GAATTC. Okay, so here CTTAAG, that means these are palindromic sequence. This also has the same sequence. So which enzyme will cut? As previously I had already shown. Okay, so EcoRI is the enzyme that can cut a particular type of recognition sequence. So here what do we add? We have added EcoRI. EcoRI will cut the phosphodiester bond that exists here. Now, say to understand this more clearly, you have to have a very clear picture of how DNA formation takes place, which is already we have talked in our previous lectures. You may refer to them. Now here phosphodiester bonds exist. So what will happen? EcoRI will act upon the phosphodiester bond which is present in between guanine and adenine. Guanine and adenine. Similarly here also guanine and adenine it will act, and here even here between guanine and adenine. So here you can see cuts DNA strands a little away from the center of palindromic sites. So it can cut little away from the center. This is the center of palindromic site, little away from the center. Okay, it will cut the DNA sequence. Now here EcoRI is acting, and so here what happened? It got cut. So the DNA sequence, it gets cut. From here the DNA sequence gets cuts at this particular location. So here it gets cut. Similarly here also it will get cut. Okay, it gets cut. Here also it gets cut. So that is between G and A. So here also between G and A. As it cuts between G and A, consequently what will happen? At this point it gets loose, and the hydrogen bonds existing here will also break down. As it got cut here, so even here the hydrogen bonds will get loose. So consequently what will happen? This portion will get detached out. Okay, so this portion has got detached out. Similarly here also it will get detached out. Okay, so it gets cut out, as you can see here. So here you get okay, here I am showing it in this manner. Okay, this portion has got separated out. Here this portion has got separated out. So here there's empty spaces left here. Here also empty spaces. These regions will be called as single-stranded overhanging sticky ends. Okay, single-stranded, because you can see here these are single now. They do not have the complementary bases. They got cut and they got separated out. Here also they got cut and separated out, leaving behind a single bases. Single bases were left behind here. So they are referred to as, we call them as overhanging sticky ends. Why sticky ends? Because they have these exposed bases, nitrogenous bases. So if they get a complementary one, immediately they can form. So now imagine this black, the one which is represented with black, comes over here. As it comes over here, then what will happen? T will get bonded to A. Again, T will get bonded to A. This T will get bonded to A, and T will get bonded to A. So what will happen? Again, they will come together. See here. Okay, so what will happen? DNA fragments join at the sticky ends. So consequently, what did we get? We got the vector DNA and the foreign DNA getting attached to each other. Okay, they got attached to each other. Then what did restriction enzyme do? Restriction enzyme detached it, cut it. Of course, here we have to use DNA ligase when we want to join them. Work of restriction enzyme will be limited to only cutting. To cutting it is limited. After it has cut it, we have to add DNA ligase. Okay, so here you see, now I'll show it to you here. Okay, just a second. Okay, so here you can see the target gene. Okay, this is the target gene where it has already been cut out. Okay, the restriction enzyme has cut it out, so you have these overhanging stretches. You can see, okay, these are the single. Okay, these are the single, the shape indicates. Okay, so here is the target gene. Okay, this is the target gene that is shown here, AATTCG. Okay, that was cut out. So here this part is removed, this part has been removed. Okay, restriction enzyme walked here, cut here, restriction enzyme cut here, and this part removed, this part. So target gene is in between. Target gene is in between. You have the long fragment of DNA, somewhere in between the target gene is there. So at both the ends, it is very important that both the ends are flanked by restriction sites or recognition sites or palindromic sites. Both the ends, if we are using an endonuclease. So both the ends should be having a palindromic sequence. So at both the ends, palindromic sequences will be cut out with the target gene, that means our gene of interest located in between. Similarly, the plasmid. Okay, the plasmid also, it will be cut. Plasmid will also cut it. So plasmid has cut it here and at the other end. So in between, it got extracted out. Okay, this portion will be loose and that will be discarded. So this got discarded. Now next event, what can be done? The target gene can be placed upon here. Okay, you can see here how it can fit. Okay, this will fit in here because you see here they are the complementary bases. These ends will be called as the sticky ends, overhanging ends. The rest, the part which got extracted out from the target gene, it will be the DNA which we had isolated, that will be removed. We will retain only the target gene containing portion, and the plasmid we will discard that small portion. We will retain the entire plasmid except that small portion that got cut out. Okay, let us see here now. So here again one more figure. So this is the foreign DNA, this is the vector DNA separately. Same restriction enzyme will cut here. So here the foreign DNA, we need this portion. We don't require the rest of it. It got cut at both the ends with overhanging sticky ends. The vector DNA, small portion, discarded this. Then next, what is done? The gene, the portion, the gene of interest will be ligated here, will be joined here. Okay, with the help of ligases, after which it will be inserted into the cells. Same way here again, construction of recombinant DNA. The fragment of DNA here, the recombinant DNA molecule, it will be inserted and then it will be inserted into the host cells. Of course, these steps we have to learn. We have not yet done. We have to learn that. So here, so as you saw here, the action of restriction enzyme, it will cut DNA strands little away from the center of palindromic sequences, leaving behind sticky stranded, single-stranded overhanging sticky ends. As you see here, the target gene, okay, that is the foreign DNA, and the plasmid, that is the vector DNA. So now the action of restriction enzyme is limited only to this. Okay, only to this it is limited. After which, of course, we can add to or we can join both of these two. But before that, so that we get more number, so that this particular step need not be repeated again and again, what is done? The target gene will be multiplied. We will increase its number. Okay, we will increase its number. Before that, we have to separate it out because now what did we get? All these things are done in a solution form. Now they have to be separated out. We do not know exactly where is our target gene. Okay, so all we will get small pieces. So those pieces have to be separated out and the targeted gene or the desirable gene selected. So that will be done with gel electrophoresis. After selecting out the target gene, desirable gene, we will multiply it. We will increase its number. Increasing its number, amplification will be done by polymerase chain reaction. These two steps I will be talking about in the third lecture. So keep in touch. Okay, I will be continuing it in the third lecture, the next two steps. Okay, thank you. And if there's any comments, any questions, then you can definitely put it in the comment box. Thank you.